cases, the short distance transport is required. Short distance transport means transport of certain substance from one cell to another cell or from one tissue to another tissue. It's for a very small distance. So when okay, there is one more student joined us. So Sevis, am I audible to you?
Yes, sir.
Okay, fine. So you are also in 12th class, right? If I'm not wrong.
Yes, yes, sir.
Okay. So we are going to start from at, like I was discussing with the Rabia. So Rabia said that this is the tough part, that plant physiology part. So what's about you? Which part it seems tough for you?
Sir, from
Sorry,
which chapter are you?
chapter. This is the like we are starting from the 11th class syllabus, plant physiology part. So
Oh,
should we start from the here? Are you okay with that?
yes, okay.
Yeah, okay. Because it's true that the plant physiology is the toughest part for the student. But for me, if you'll ask me, this is the genetics which is the toughest part in when you read the genetics from the point of view of this one medical examination. You know, do you know? Okay, one more important thing which I want to discuss here. So, have you any idea, Ravi and Sevis, that which part is the most important or which part have the maximum weightage in your medical examination in biology,
Physiology.
zoology? What about you, Rabia? Which part have maximum weightage? Any idea? Yes, Rabia.
I don't have any.
Okay, fine. So first, before starting the class, let me know you. As per my own analysis from the last 10-year question paper, I have gone through with that and I have made a list of these chapters. So I'll give you a clue over here. I'll give you an idea over here that what is the weightage and which part. The maximum weightage goes to the genetics part. Genetics part you can't imagine. Genetics part you may get question uh, minimum you'll get 18 questions and maximum 20 questions. So you can imagine that the weightage is of 80 marks, right? So 80 marks is the weightage for that. Then second number from the genetics, then it comes to the biotechnology. Biotech part. From biotechnology part, you will get almost 8 questions, right? 8 questions. Biotech part from then which part is very important? Then it's come reproduction. Reproduction, when I say reproduction, we include both of the part, that plant reproduction and human reproduction. Plant plus human. Both kind of the reproduction. Reproduction and reproductive health. Then fourth number, that is the plant physiology and animal physiology. Plant physiology have less chapter, but plant physiology, you will get 10 questions from here. And equal questions are being asked in human physiology. So human physio also have human physiology. It also have almost 10 questions. Means 10, 40. So total 40 marks weightage. Sixth number, it comes to the cell biology. Cell biology, it contains cell cycle and cell structure both, right? Biomolecule, hardly you will get one question. So biomolecule is that not that much important for your this. Then what about you? Then seventh one, that is the anatomy part. Anatomy plus morphology of plant. Morphology of plant. Then comes to the rest of the chapter, right? So this is the sequence of the weightage, and that's how the weightage have been decided, right? So this is my analysis, and we have to study, right? We should study like this way. But right now, this is for the dropper student. So as you belongs to the 12th class student itself, so genetics you will go through with it, right? Genetics you will go through with biotech, you will go through with it, right? if you would have been in like you would have completed your 12th class, then I might have started from the genetics section. But right now you are in the 12th class, so first we should complete the 11th class syllabus, right? So from 11th class syllabus, plant physiology is a bit tougher part. Animal physiology, I know that human physiology is a bit easier part. So you're right, Rabia. Plant physiology is a bit tough until unless you understand it. But believe on me, when we will complete this part, then you will feel more confident, right? Okay, so let's start from one by one. You will get the link of this PPT from whoever have contacted you and who are like coordinating from our side. So he or she will give you the link of this part. So you need not to note down everything, but whatever I dictate during our conversation, whatever I dictate, that you have to jot down. Fine. Okay. So I was telling that transport in the plant transport of material. So normally two things are transported in the plant inside, right? What are those two things? The first thing is that is the water and minerals. So water and minerals, they are transported from the root to the top of the plant, right? Second thing which need to be transported, that is the organic substance. Basically, this organic substance is a food, right? Then food transport. Along with that, there are few substances which need to be transported within plants. What are those? They are the hormones, basically, phytohormones. We'll study those phytohormones. Auxin, gibberellin, cytokinin, ethylene, abscisic acid, and all that, right? They need to be transported. So water and food, it need to be transported for the long distance. Why? Because if a plant is of 10 meter, then water have to travel 10 meter from the root to the top of the plant. And again, for the food, if it is 10-meter plant, then food have to travel from the leaf to the downwards 10 meter to the root, because root cannot make their own food. They are dependent on the green part of the plant, right? So these are the long distance transport. Then what about the short distance transport? So I told you there are two kinds of the transport in the plant: short distance transport and long distance transport. In short distance transport, these are the hormones. So hormones are transported in the short distance, right? Phytohormone. So how does the short distance transport takes place? First, let's start with the short distance transport. Short distance transport means transport of certain substances like phytohormone from one plant cell to another plant cell. It takes place by means of two things. There are two means: diffusion and osmosis. These are the two means through which the transport of substances takes place. We will discuss about the diffusion and osmosis. Let me brief you. Diffusion is transport of molecule, or that is a movement of molecule from its higher concentration to the lower concentration. This is called diffusion. Second thing is osmosis. Osmosis is transport of the water from its higher concentration of water to the lower concentration of the water through semi-permeable membrane. Right. This is, I am just briefing you right now. We will discuss in detail that what osmosis exactly is. Okay. For short distance transport, so one more important. This is a plant cell, right? Plant cell have plasma membrane, and around the plasma membrane there is a cell wall. One more layer is present. This one more layer which is present around the uh, this one. This is called, what we call this? We call it, what we call it? That is cell wall. This is basically a cell wall, right? So this is cell wall, and below the cell wall, beneath the cell wall, there is a plasma membrane, which is also called plasmolemma. And then there is a protoplasm. Protoplasm is peripheral. It is found in the peripheral part. Why? Because, and nucleus is also peripheral. Why? Because the most of the part of the plant have been captured by organelle called vacuoles. So vacuoles are so they are basically called central vacuole or vacuole. So central vacuoles are the largest organelle of the any plant cell, right? Central vacuole, they cover the largest area of the plant cell. What happened? These central vacuole or these vacuole, they store water, they store iron, and they store the food, right? So transport of substances takes place from one vacuole to another vacuole. How does this transport takes place? Basically, there is a connection or there are some pores between two cells. These are called plasmodesmata. So transport of substances from one cell to another cell in the short distance takes place through the plasmodesmata, and means of the transport can be either diffusion or osmosis. Is that clear? Short distance transport.
Yes, sir.
Yes, sir.
Okay, so please note down one thing. Note down. Have a notebook with you. Transport of substances between two plant cells. Transport of substances between two plant cells takes place through plasmodesmata. Full stop. These substances are transported. The substances are transported from one central vacuole to another central vacuole, from one central vacuole to another central vacuole by the means of diffusion or osmosis. By the means of diffusion or osmosis. This was something about the short distance transport, right? Now this is the long distance transport. First, I want you to please understand this concept. Let me tell you this concept. This is a basic concept and how the transport is done, right? Okay. Look, transport of so this is the leaf part. So leaf part, long distance transport will take place of two substances. I have already told you. First thing, that is the water and mineral, and second thing, that is the organic substance or food, right? Let's talk about these both of the things. So transport of water. So basically leaf require water for the photosynthesis and plant cell require water to maintain its shape. So there are the requirement of water is for two purposes. First, for the transpiration, second, for the photosynthesis, and third, for maintaining the cell shape, right? If the water will be scarcity of water will be over there, then what will happen? The cell will change its shape, right? Look, there are certain pipeline. These pipeline are known as xylem element, right? Xylem element, they transport the water from where water and mineral are transported by means of a tube or pipeline. These pipelines are known as xylem element. Xylem element are a type of complex tissue. They are a type of complex tissues, right? Complex tissues, we know that they are made up of more than one kind of the cell, right? So these xylem transport water and mineral. Water and mineral is absorbed from the soil, right? So this absorbed water and mineral, they are transported through the xylem to the leaf, right? Then what happened? Xylem element is a compound tissue. So it is made up of more than one kind of the cell. So what are those cells? It is made up of basically four kind of cells: trachete, vessels, xylem parenchyma, and xylem fiber. These four kind of the cells make a xylem tissue, right? So how? So right now we know that how, like which tissue transport water and mineral from the root to the top, but how it is transported. So this mechanism of transport, right? How it is transported, or by the mechanism by which it is transported from the root to the top, it has been explained by a theory, and that is known as the transpiration pull theory. We will discuss that theory. So transpiration pull theory is a theory in this chapter that will explain how the water is or mineral is being transported from the root to the top of the plant, right? There are another tissues. These tissues are known as the phloem, right? Phloem, now leaves make the food. So leaf have taken water and mineral from the root. Now this is the responsibility of leaf to give food to the root, because root are not able to make their own food, right? If they will not supply food to the root, root will die, and they won't get the water. So this is a mutual understanding. This is the mutual benefit. It's a type of mutualism. Both are helping each other, right? So in return of water and mineral, leaves are giving back root food, right? So formation, one very important thing. Food is formed. Food is formed in the leaves, right? By the process of photosynthesis, right? Food is formed. This food is transported to the root with the help of a tissue called phloem, right? Phloem transport organic substances and food by the phloem, right? And phloem is also a type of compound tissue, right? Means phloem will also be made up of more than one kind of the cell. So phloem is made up of four kind of the cell: tube cell, companion cell, phloem parenchyma, and phloem fiber, right? So now we came to know that transport of organic substance from leaf to the root takes place through the phloem. But how it is transported from the leaf to the root? So there is a theory, that theory known as mass flow theory or cell and sink theory. This theory explain, or this mechanism explain it. This theory explain the mechanism by which food is transported from the leaves to the root. Right. So there are two theories: transpiration field theory. It explain how the water is being transported, and mass flow theory. It explain how the food is transported. Now I have a question. Right. In which form food is transported from one point of the plant to another plant? Means from leaf to the root, food is transported. What is that form? Tell me. Who will tell me? Rabia and Savis, this question is for both of you. Tell me.
Sucrose.
Very good. Excellent. So, look, formation of food takes place in the form of glucose. Glucose is monosaccharide, right? Now, the glucose is converted into sucrose, and transport of, so formation of food takes place in the form of glucose, but translocation, right, means transport of the food takes place to the sucrose. Is that clear? Is that part clear for both of you? If you have doubt, please ask.
Yes, sir.
Great, great. So now let's start with the other processes, how the how the food and water is transported. So means of transport. So there are different means of the transport. So basically, there are two type of the transport in the plant, right? What are those two type? Active transport and the passive transport. Now, what is the passive transport, right? So we know that there is a process of diffusion, right? Diffusion I have explained over here. Diffusion is transport or movement of molecule from its higher concentration to lower concentration, right? So diffusion is a very easy process, right? We just open a bottle of deodorant, and what happened? A deodorant start diffusing from its higher concentration from inside the bottle to outside, from higher concentration to lower concentration. So they same thing. So energy is not required for the diffusion, right? And when any molecule transported from its higher concentration to lower concentration, energy is not required, and we call it downhill transport, right? Or we call it transport of molecule in the direction of concentration gradient, right? So when any molecule is transported from its higher concentration to lower concentration, we call it downhill transport, or transport of molecule in the direction of concentration gradient, right? Now, suppose that if this process have to go opposite, right, means in naturally without energy, the transport is taking place from its higher concentration to lower concentration. But now we want, in certain cases it happen. So now we want transport of substance in opposite direction, means from low direction to low concentration to higher concentration. this is called, here energy is required, right? And this is called uphill transport. And now the transport will be against concentration gradient. I have written over here, right? Now this is called uphill transport. And now the transport is against concentration gradient. When you move against concentration gradient, against the natural direction, then you have to spend the energy. And the same thing is happening over here, right? When the movement of substances will take place against the concentration gradient, we have to spend energy. When it will take in the direction of concentration gradient, right, we do not need to spend the energy, right? So look, in active transport, first let's talk about the passive. In passive transport, it is always downhill, means it always takes place in the direction of diffusion gradient, from higher concentration to low concentration, and no energy is required over here. Active transport, it is uphill movement, means this transport takes place uphill, means in the always opposite to the diffusion gradient. It always takes place from from a low concentration to higher concentration, and here ATP is spent, means energy is spent. For example, transport of mineral. So transport of mineral from soil to root is always active transport. Why? Because soil have root have always more concentration of certain minerals than soil, but still it require more minerals, so it will always be active transport. Clear? Active passive transport.
Yes, sir.
Yes.
Great. Now diffusion can be, now how the diffusion takes place. So diffusion, we know that is a passive transport, and diffusion takes place through, first going, before going to the diffusion, we need to understand the structure of plasma membrane, right? So I want to ask you a question. There are three type of the permeability. First, permeability, then semi permeability, then selectively permeability. So what is the difference between these? Permeable, semi permeable, and selective permeable. Anyone of you? This question is for both of you. What is the difference between these three? Permeable, semi permeable, and selective permeable.
Selectively means for selective compounds.
And what is semi permeable?
In semi-permeability, some like few substances are allowed to pass through, and few substances are not allowed to pass through.
Right. Then selective.
In selective, only selected substances are to pass through.
Right.
Other than that, no substance.
Right. Very good. So let's make it more clear. Right. Look, you know, outside of this building there is a public road, right? So there is a gate, a very big gate, and it's a road. So anybody can pass through that gate. That is a permeable. So that gate is permeable. Anybody can pass through because that is a public gate. So I will say this road is permeable. Outside of this building compound, there is a gate. Again, another gate. Inside that gate, only those people who can come, who have to do some job with this building, right? So this is, and one more thing, truck and big vehicles, they cannot pass through this gate, right? Only certain size and certain type of the people and certain type of the vehicles can pass through that, right? Because it's not a very big gate, right? So this gate is, now this, that gate was permeable, now this gate is semi permeable. Certain size of vehicle can come inside, certain type of vehicle can come inside. Now within this office, only particular specific type of the people can come. Either they will be teacher, or they will be student, or they will be from some our team or coordinator or office work. So there are 4 and 5 kind of the people can come inside this office. No one else compared. So I will say the gate of my office, that is a selective permeable. The same thing happened with the plant. In plant, the cell wall is permeable. Almost all kind of the thing can cross the cell wall. When I talk about the plasma membrane is semi-permeable, right? Only certain size of the substances can pass through this plasma of particular size, not more than that size, right? So only particular size of the substances can pass through. They have, if they have a particular, they are of particular size, then only those kind of substances will pass through. So this plasma membrane is semi-permeable. Now come to the nuclear membrane. Nuclear membrane is selectively permeable. Only those substances or those compound will go inside the nucleus which is required, and those which is not required, they cannot go inside, right? So that is the difference between permeable, semi-permeable, and selective permeable. So always remember, cell membrane or cell wall is a permeable, plasma membrane is semi-permeable, and nuclear membrane is selectively, right? So before starting diffusion through the lipid and protein, let's talk about the plasma. So plasma membrane have now got a latest term that is called plasmolemma or biomembrane. So we most frequently we call biomembrane. Why? Because all the biological organelle, if they are surrounded with any membrane, that will be the plasma membrane or biomembrane, and the composition will be almost same, right? So plasma membrane is made up of two things. What are those two? Bilayer of lipid. Look here, bilayer of lipid. So there are two layers of lipid and protein molecule, right? These protein molecules are called channel protein, right? So bilayer of lipid. So this is the lipid molecule. Try to understand. This is the lipid molecule. Lipid molecule have a head and a tail, right? Tail always remains toward the center, and head always remains toward the outside, means outer surface. So try to understand. Head is hydrophilic, means it is water loving. It can make the bond with the water. But the tail part of lipid molecule is always hydrophobic, water repelling, right? So tail always remains toward the center and head always remains toward the outside, right? Okay. Now there are certain there are protein, they traverse across the membrane. These are called channel protein, right? Channel protein can be of two types. Try to understand, please. Try to understand. And if you have any doubt, please you have to ask me. Look, now the channel protein, I am discussing the structure of plasma membrane. So plasma membrane is made up of two things: bilayer of lipid and a protein molecule, right? And protein, sorry, and protein. Protein, so those protein molecule will traverse across the membrane. They are called channel protein. Channel protein can be of two types. If they transport water, they are called aquaporin. If they transport ion, they are called ion channel, right? So there are two kind of the channel protein. This thing you need to understand, right? You will, uh, the most frequently asked questions are from this part, right? Now, what will happen? All these substances. So this is the composition. Now try to understand. Diffusion through lipid. All lipid soluble substances. Question have been asked many times from this point. All lipid soluble substances, they are transported, they are diffused directly through the lipid, right? They are hydrophobic substances and they are fat soluble substances. So fat soluble substances, hydrophobic substances, or lipid soluble substances, they directly diffuse to the lipid membrane, right? From higher concentration to lower concentration. But water soluble substances cannot cross this lipid bilayer, in spite of there is a, they want to diffuse in the direction of concentration gradient. So what will happen? They will pass through the hydrophilic substances, means water loving substances, water soluble substances. They will transport it from higher concentration to lower concentration through these channel protein, right? So channel protein, so all hydrophilic substances, water soluble substances, they are passes through the channel protein, right? And all lipid soluble substances transported to the lipid bilayer. Is it clear or not? Tell me. Clear?
Yes.
Both are the passive. Rabia, is that clear?
Yes, sir.
Great. So you need to remember aquaporin. Aquaporin have been asked multiple time, many times. You medical question. This is the medical question. Lipid soluble substances can pass through. This question have been asked. Channel protein can be off. This question have been asked, right? So again, the channel protein, this one, channel protein uh, are of they diffuse the substances by two ways: simple diffusion and facilitated diffusion. Try to understand. Channel protein diffuse substances by two methods. First method is simple diffusion. In simple diffusion, like you can see, the substance can cross through this channel protein, right? Simply, this is a channel protein. For example, this is a channel protein. So, any substance which have to pass through these channel proteins, simply the substance, like suppose that this is the sodium ion. So simply it will be transported from one end to another end. It will go inside or it will come outside. So this is simple diffusion. And it will be in the direction of, because both are the what? Diffusion is obviously a passive transport, right? Now in facilitated diffusion, what is that? In facilitated diffusion, this is also passive, energy is not required, but substrate means that substance which need to be transported, that will be taken or received by a protein, and that protein will pick it from one end and drop it at the another end. These channel protein have a special type of the protein that is called carrier protein. So in facilitated diffusion, carrier protein is required, which will pick from one end and will drop at another end. But in simple diffusion, the carrier proteins are not required. Is that clear or not? Tell me.
Yes, sir. Clear.
Yes.
Clear? Okay. So remember this point, this part was very important, right? So facilitated, you need to what you need to remember that facilitated and simple diffusion both are the do you have NCRT with you or not? Do you have your NCRT with you, both of you?
The textbook.
Yes.
Yeah, textbook, right.
Yeah, sir.
Okay, keep it with yourself, right? Uh, because note it down. In page number 117, there is a line, and that line have been asked twice as a question. Please note down. There are there are 8 different type of aquaporin. There are 8 different type of aquaporin in the cell membrane. The question have been asked how many type of the aquaporin, and this question was directly from your textbook, right? From page number 117, there is given in the third paragraph of this page, right? It is given, and the same question, the same line have been picked. So some questions, like most of the questions, 70% question you will get from your NCRT, but 30% or 25% question, they will be not directly from the NCRT. There will be some peripheral area of the NCRT. Exactly, they have not been mentioned in the NCRT, right? But you will get questions. So I'll keep on telling you those parts, right? So this was simple diffusion, facilitated diffusion, right? What are the aquaporins? What are the channel protein? And it's clear to you. That's very good. So now come to the passive transport. Even from this part, the question have been asked. So I have already told you, passive transport takes place through the lipid membrane, bilayer of lipid. So diffusion of lipid soluble substance. Then facilitated diffusion takes place also in the passive means, right? It requires carrier protein, and active transport energy is required. So one more thing, it's very important, right? What are the pump, right? Please note it down. The channel protein, the channel protein which transport the channel protein which transport substances, the channel protein which transport substances by means of active transport, by means of active transport are called pump. Right? So these are called pump. Now channel protein. So the same thing I have tried to mention over here. So channel protein can be of three types. Basically, they are of two types: uniport and co-transport. In channel protein, when in one time only one type of the ion or one type of the substances are transported, this is called uniport. Suppose that this is a channel protein. There are hydrogen channels, right? Hydrogen channels, right? So hydrogen channel, they will transport only the hydrogen one time, or sodium channel, they will transport only sodium ion. So this is called uniport, either in inside or outside, right? In any direction, inside or outside, only one time, only one type of the ion can be transported. These are called uniport. There are symport or co-transport. Sorry, co-transport. In co-transport, simultaneously two ions will be transported, either in same direction or in opposite direction, right? So in co-transport, two type of the ion will be transported simultaneously. But co-transport can be of two types: symport or antiport. In symport, both of the substances transported in the same direction, but in antiport, both of the substances are transported in opposite direction, right? So please note it down. Symport are found symport are found in a small intestine. This question have been asked in AIMS. The symport are found in a small intestine, like fructose and sodium. Fructose and sodium are absorbed. Fructose and sodium are absorbed simultaneously in same direction. This is the best example of symport. Next point: sodium and potassium pump. Sodium and potassium pump. Sodium and potassium pump are found in the membrane of sodium and potassium pump are found in the membrane of neurons. They are the best example of antiport. Full stop. They transport 3 sodium ion and 2 potassium ion. They transport 3 sodium ion and 2 potassium ion in opposite direction. So that is the channel protein can be of three type, or channel can be of three type: uniport, symport, and antiport. Is that clear? Remember both of the example.
Yes, sir.
Okay. Remember the example of digestion. Remember the example of, that's why I have mentioned this example. Both example I have been asking you a particular question. So that kind of the question you will get. Impermeable, permeable, semi-permeable, selectively permeable. I have told you, already explained.
Sir,
Yes.
if you don't mind, can you repeat the point of the transport 3 sodium ion? That
Okay, fine. The membrane of neuron, we'll discuss ionic exchange theory in that neural system again, right? The membrane of neurons have sodium and potassium pump. The membrane of neuron have sodium and potassium pump, full stop. 3 sodium ion and 2 potassium ion, 3 sodium ion and 2 potassium ion are transported in opposite direction simultaneously through these pump.
Sir,
Yeah.
the channel protein and pump are different.
Yes, channel protein and pump. Look, pump term is used for basically those protein, transport where it is active transport. And normally in the passive transport is done through the only channel proteins.
Okay, thank you.
So pump are, we can say like this way. Pump are a special type of the channel protein which transports substances actively. Clear?
Yes, sir. Active transport.
Exactly, active transport. And what about the this one, facilitated diffusion? It is done through the
Channel protein.
no, passive transport. Passive transport. It is done
Special
facilitated. It is done through the
Facilitated.
aquaporin, right? Aquaporin and
Ion.
carrier protein, right?
Yes.
Yes. Rabia, are you asking something?
no, sir, no, sir.
Okay, fine. Osmosis. Osmosis is a very simple concept, but it's very confusing kind of a concept, right? So let's remove the confusion from here. Try to understand first. Focus on the PPT. Look. So suppose that I have 2 tumbler, right? So in one tumbler, so this is tumbler A and this is tumbler B. In one tumbler, I have 1,000 milliliter of water and 50 gram of the salt, 50 milligram of the salt. And another tumbler, I have 1,000 milliliter of water and 100 milligram of the salt. So the salt concentration is low here in A cell. So I will call it, this is low concentrated solution. So I will call it hypotonic, right? And this is higher concentrated solution. So I will call it hypertonic solution, right? So suppose that, try to understand me. Suppose that I connect them, both of I connect them through any means, right? Direct connection. So what will happen? Salt will start moving from hyper concentration to lower concentration. Means diffusion of the salt will take from hypertonic to hypotonic, and diffusion of water will take from hypotonic to hypertonic. Is that clear?
Yes, sir.
I have a example, and with the help of that example, I want to ask you a question. Suppose that I brought a marine fish, right? I brought a marine fish. It is a small scoliodon shark, and I put it in a river water or a soft water, a sweet water, on a big aquarium that is filled with the sweet water. What will happen with that fish? I have a marine fish, and I have put into a different environment that is a soft water or sweet water. Any idea?
It will die.
Definitely it will die, but how it will die? That is a question. Why it will die?
Because it is used to that marine and salty water. Now that it's in fresh water,
Okay.
there's no salt. There's no concentration of salt in the fresh water, so
You're close to the answer. Shavis, any idea? Look,
No.
yes. Look, the fish is living in, try to understand my concept. The fish is living in marine water, right? So the concentration of the water and concentration of the blood always will be same, right? So fish is living in high concentrated hypertonic solution, marine water, salty water, hypertonic solution. So blood will be hypertonic. Now you have kept that fish in hypotonic solution. So what will happen? Water will start moving from hypotonic to hypertonic. Means water will start moving from outside to inside the fish. Fish will get swell. Its body will swell and it will die. And the same way, if I keep a freshwater fish in a marine water. Now the blood of freshwater fish is hypotonic, right? This one, hypotonic, and the external environment is hypertonic. What will happen? Water will start coming from hypotonic to hypertonic. Means water will start coming from blood to the outside. Fish will shrink and it will die. Did you got my concept?
Yes, sir.
Yes.
Same thing like you put a raisin in the fresh water and raisin swell in the morning. You put a grape in the salty water and grape shrink. The same thing happened with the fish, right? So now come to the osmosis. In diffusion and osmosis, there is only one difference, that in osmosis, there is a semi-permeable membrane, or this is also known as the differentially permeable membrane. Semi-permeable membrane or differentially permeable membrane have a small pore, very small pore. These small pore have a certain size, so that only a certain size of the substance or certain size of a molecule can cross through this membrane, right? So when two substances are, so suppose that this is the water molecule and this is the sucrose molecule. So this is the solution. So try to understand. This is a solution, right? This sucrose solution have high concentration, right? And this water molecule, this is hypotonic, right? So this is hypotonic, I have written, and this is the hypertonic. Now we have separated them with the help of, so suppose that there is concentration is 10 millimeter Hg, right? That is the pressure, and here it is 4 millimeter Hg. That is the diffusion pressure, right? So what happened? Now, this sucrose molecule cannot cross through this semi-permeable. Otherwise, sucrose molecule would have gone from the cell number 2 to cell number 1. But this is not possible because say they cannot cross through the semi-permeable. So what will happen? Water will start moving from hypotonic solution to hypertonic solution until at the both sides the concentration become equal. The water will keep on flowing, right? So we can say high concentration of water, movement of water from high concentration of water to low concentration of water is called osmosis. Movement of water from hypotonic solution to hypertonic solution through semi-permeable membrane is called osmosis. Or movement of water from low concentrated solution to high concentrated solution is called osmosis. So you can define osmosis in three ways, right? So this is the osmosis. And uh, this reaction will keep on going until the concentration become equal at the both side, means until the equilibrium is achieved. Is that clear to both of you?
Yes, sir.
That this question, when the question come, you will get the question. I will give you the question paper. We design the question paper like biology question. I just make the biology question, not team member. They just type and they arrange the they make the question paper. They just type the questions and they make the sequence and formatting, all that. But question paper, I'll design the biology question paper. Your physics sir and chemistry sir will design the chemistry and respect to their question paper, right? So I'll give you this kind of the question, and I'll give you some question which have been asked in previous year question paper. So then you will come to know that this question is very simple, but it is very confusing because sometimes they define it with in terms of solution, sometime in with the water, sometime with the concentration. So it's very confusing. So that's why I have written all these things in the same row, right? Now, what is the DPD? Diffusion pressure deficit. Diffusion pressure deficit. So, very important, try to understand. So, what is the diffusion pressure? Diffusion pressure is a pressure which is suppose that these water molecules are passing through this membrane. So they are this water molecule, the red color show it's the arrow shows the movement of water molecule. So when the movement of water molecule will take place from higher concentration, low concentration to higher concentration, right? Solution. So these water molecule they have the more free energy, right? So they will impart a pressure on this semi-permeable, and that pressure will be termed as what? Diffusion pressure. Clear? So look, pure water have means low concentrated solution have more diffusion pressure, and high concentration solution will have low diffusion pressure. So here the diffusion pressure is 10 mmHg. Let's say here the diffusion is 10 mmHg. Here the diffusion is 4 mmHg. So how much more diffusion is required? How much deficit is there? When the diffusion pressure will become equal 7 at both of them, then this process of diffusion will stop, right? So here it is 10 mmHg, and what should be? It should be 7, means there is 3 extra, and here it is 4 mmHg. What should be? 3 more, right? So here 3 mmHg diffusion more required. So what is the deficit? The deficit is 3 mmHg. So the water will start moving from cell 1 to cell 2 until it achieves the equilibrium. So we can say 3 mmHg is DPG, diffusion pressure deficit. That much diffusion pressure is deficit, and when the water will come over here, the diffusion pressure will develop and it will become 7 at the both of them. So this is called DPD. Tell me, is it clear or not? DPD is clear or not?
Sir, can you just say once
Sir,
again?
if you don't mind, can you explain it again?
Yeah, sure. Try to understand. So first, I want to tell you what is diffusion pressure, right? So diffusion pressure, pure water have more diffusion pressure. Diffusion pressure is free energy of water molecule, right? So look, here the water molecule are in pure state. They are free. They freely collide with each other. So there is more free energy, right? Means there is more diffusion pressure. Now come to the cell B, cell 2. In this cell 2, what happened? The sucrose is present over there. Water molecule are colliding with the sucrose, means they will lose their energy. They will have less energy. So here diffusion pressure is less. Why? Because salt or because sugar molecule is present over there, right? So diffusion, let's for example, here in cell 1, diffusion pressure is 10 mmHg. Here 4 mmHg. If diffusion will start, when diffusion will stop? Diffusion will stop when at the both side the pressure will be 7 mmHg. That will become equilibrium, right? To achieve 7 mmHg, how much pressure is less over here, this side in concentrated solution? How much pressure is deficit? How much pressure is less? Tell me.
3.
3 mmHg,
3.
right? So I will say 3 mmHg is diffusion pressure deficit. Got it? DPD.
Yes.
So we can
Yes,
say
sir.
movement of water molecule take place from high DPD to low DPD. Clear?
Yes.
This is the same thing. This is the osmosis. But now we are explaining the osmosis in terms of physics. Here we have explained the osmosis in terms of chemistry. Here we are explaining the uh, movement of molecule with the angle of physics. That is the difference. So DPD is a physics term, and this one osmosis is a chemistry term, right? So, but the both concepts are given in your NCRT. That's why I'm explaining, right? Very, very important, very easy question. The number of time the question have been asked with the help of this diagram. You can get this diagram. So diagram based question have been asked, or the question have been asked differently, right? So look, what is the turgor pressure? What is the wall pressure? What is osmotic pressure? So this is a cell. It has been kept in a water or pure solution. So water is moving from outside to inside. Means from high osmotic pressure, right? From high osmotic pressure or high diffusion pressure to low diffusion pressure, right? Means from high osmotic pressure. So movement of water always takes place from high osmotic pressure to low osmotic pressure. This is called OP. When water will get fill inside the vacuole or vacuolar sac, what will happen? The cell will turgid, means it will swell. So we use term when cell will swell, we will call that cell is turgid. When cell will turgid, what will happen? This will start putting pressure on the cell wall. So due to turgidity of cell, due to swelling of cell, the pressure is imparted from inside cell to outside on the cell wall. This is called turgor pressure. You have a balloon, you are blowing air and filling balloon with air. Air will impart a pressure on from inside. Air will impart a pressure on the wall of balloon. You will call it turgor pressure, TP. Now what will happen? The wall will put pressure inside, right? Again, the turgor pressure, wall will develop a pressure from outside to inside. This is called WP, wall pressure. And WP is always equal to TP. Means wall pressure will be always. So cell wall will try to maintain the shape and it will make a pressure from outside to inside against the turgor pressure or TP, right? So wall pressure will be always equal to TP. Is that clear?
Yes.
Yes, sir.
This formula should be clear to you and you have to remember. DPD, diffusion pressure deficit, is equal to OP, means osmotic pressure minus wall pressure, right? And we know that WP is equal to TP. Wall pressure is equal to TP. We can also write OP is equal to DPD is equal to OP minus TP. Is it clear?
Yes, sir.
You have to remember this concept. This diagram, the question have been asked in medical examination from this
Sir,
diagram.
can you repeat?
Yeah, sure. Look, there is a formula. DPD, diffusion pressure deficit, is equal to OP, osmotic pressure minus wall pressure, right? Both pressure are in opposite direction, right? OP minus WP. But we know that wall pressure is equal to TP, so we can also write DPD is equal to OP minus TP. Is that clear?
Yes, yes.
Okay. There is one more bit complex, mm, a bit complex concept that is called water potential. Now, what is the water potential? Water potential is free energy of water molecule, right? Free energy of water molecule. Water molecule have some free energy. Because of this free energy, they collide, right, here and there. So that is represented by, and what potential is represented by psi, psi, psi term, right? And psi W is water potential. And what is water potential? Water potential is free energy of water, right? So high water concentration means high water potential, right? Low water concentration means low water potential, right? A pure water will always have water potential 0. So pure water will always have water potential 0. Is that clear? Even this question I've been asked, but it is very older question. You won't get that much simple question nowadays, right? So if you will add some solute, what will happen? The water potential will decrease or not? Decrease. So
Yes,
it will
sir.
go into minus. So while
Yes.
adding the solute, try to understand, while adding the solute, so suppose that I have 100 milligram of water. I have added 2 gram of sugar in this. So the water potential will become negative. So suppose that it become -2. So I will call it solute potential. So what is the solute potential? After addition of the solute, the amount of water potential which decrease, or the value of water potential which decrease, that is called solute potential. Right?
Yes.
So we represent it with the psi s. What is psi p? Pressure potential. Right? So when you will add on some solute, right, what will happen? Pressure potential will, means pressure of the water will decrease, right? So that is called pressure potential. So there is a formula: water potential is equal to solute potential plus pressure potential. This is the formula. Tell me, is it clear or not? Then I will go ahead.
Yes. So what is this pressure potential?
Pressure potential is, look, suppose that I have here it's a pressure, right? Here, suppose that there is a pressure of 7 mm Hg, and here it's a pressure of 10 mm Hg. So when I will add on more salt, what will happen? This pressure potential, means free energy will go down, right?
Yes.
So free energy, the amount of free energy which will go down, right? That will be called pressure potential,
Okay.
right? Look, again, I have tried to show with the help of example. Try to understand. So maximum free energy will be in pure water molecule, right? Free energy means chemical energy or kinetic energy, right? So water potential, psi W, water potential of pure water is 0, right? Okay. So suppose that in this solution, there is a psi W is -2, right? When we add on the solute over here the psi water, means water potential is -3. So what will happen? Water will start moving from its higher potential to lower potential, right? So addition, this glucose I have added, solute addition. So when I will add the solute, psi W, solute potential will become -2, right? Because due to addition of solute, what happened? The water potential got down, means chemical energy decrease, right? And water potential will become negative. Water potential will become here -2, right? So whenever we will add, so psi p is present thing, psi p is pressure potential, psi is solute potential, psi w water potential, right? And to get the water potential, we have to add both psi s and psi p are the same. And what is the potential? Means the free energy or chemical energy of the water. Hope it should be clear to both of you. And if you have any doubt, you can ask me.
Yes, sir.
So, sir, pure water will have more potential.
Sorry, yes, pure water will have maximum potential. And that what is the maximum value? 0.
Free energy.
Yeah, free energy, right? Maximum free energy. Pure water will be having maximum free energy, and that will call psi w, and that always be 0. This question I have been asked many times, right? And when we will add on the solution, we will call it solute potential, and when we will add on the solute, the water potential have to go down, means the value will be negative. So water potential, one more thing, water potential can never be in positive value, never ever. Clear?
Yes.
Yes, sir.
Even the question have been asked from here. Stages of plasmolysis. So what is the plasmolysis? So suppose that I have given an example. Suppose that I have a grape, and in that I have a grape and I have a glass of water, and in the glass of water, that is a concentrated salt solution. When I will put a grape on here, after few hours, what will happen? Grape will shrink. So what will happen? The water of grape will come out. Means the water will come out from the hypotonic solution to hypertonic solution, right? So this is called plasmolysis. The same thing happen over here. So there are 4 stages of plasmolysis: limiting plasmolysis, incipient plasmolysis, evident plasmolysis, and final plasmolysis. So try to understand all these stages of plasmolysis. Look, Suppose that these cells have been kept in hypertonic solution. So this is a flaccid cell, normal cell. What will happen? Plasmolysis will take place, and during this plasmolysis, water will start coming out from vacuole to the outside of the cell, right? So water will start coming out from hypotonic solution to hypertonic solution. This is called plasmolysis or limiting plasmolysis, right? Again, in second, incipient plasmolysis. What will happen in incipient plasmolysis? Suppose that we increase the external concentration more, means we make it more hypertonic solution, or the cell is kept for the longer time period. Now more water will come from the central vacuole. Now the protoplasm or plasma membrane will start pulling itself from the corner of the cell wall, right? So space will be there between the plasma membrane. Plasma membrane is coming out of the corner, and this space will get filled with the hypertonic solution. This is called, when it will leave the corner, when plasma membrane will leave the corner, this will be called incipient of plasmolysis. Then next stage is evident plasmolysis. In evident plasmolysis, the plasma membrane or protoplasm will be in contact with the cell wall at very few places, and it will leave from the most of the places, right? This is called evident plasmolysis. In incipient final plasmolysis, more water will come out of it, and protoplasm will leave the cell wall, and hypertonic will get filled between the space between the protoplasm and the cell wall. This is called final plasmolysis. One last thing, if we keep this cell is still alive. Final plasmolyzed cell is still alive. If we will keep this cell in normal water, what will happen? The cell will again regain its shape and become alive. But this is possible that if it is kept immediately after plasmolysis, if it is kept in the normal solution or pure water, then this is possible, else the cell will die. So tell me, is it clear? The question I've been asked many times from this concept. What is the incipient plasmolysis, or what is the evident plasmolysis? That kind of a question. What is the limited plasmolysis? And there will be four statement, and you have to define which is the correct statement or which is the correct concept. Tell me, both of you, Saviz and Rabia, is it clear?
Yes, sir, clear.
Yes.
Okay. So the same thing I
Can
told
you just say what is incipient plasmolysis?
Sorry, incipient plasmolysis. In incipient plasmolysis, only the protoplasm will leave only the corner from the cell wall. That's it.
Okay, sir.
But it will remain
And
connected
water
in
will
the
be stored there.
only corner. Yes. But in evident, it will leave all the point only except very few point. It will leave all the points. That is the difference between incipient and evident. Clear?
Yes.
What is imbibition? When any solid substance absorb water or any solution solvent, that is called imbibition, right? In for example, we have a gram seed, right? And we soak them seed over the night with the water. What will happen at the morning? This solid seed will absorb the water. That is called imbibition. When any solid surface absorb the water, this is called imbibition. Is that clear?
Yes, sir.
Okay. So keep it till this point. So
Sir, can I repeat once again?
yeah, sure. Ambibition is the process when any solid surface absorb water or solvent.
Okay, sir.
For example, soaking the seed in water. That is ambibition. You must have seen during the rainy season, your door and wooden window become tight. That is ambibition. They absorb the moisture from their outer surface. That is ambibition.
Okay.
So that's it for the day, right? In the next class, we'll start from the from the next topic, like the absorption of water on wood from here, and then the root pressure and all that from transport part. So before summing up the class, I want to know, so are you okay with the pace which is going on, or should I make it faster or slower? Tell me, first thing, both of you, Rabia and Savis.
This is good.
This pace is okay,
Yes.
Rabia?
Yes.
Okay, fine. So do you want something more? Do you want me to dictate anything or something else? If you have, otherwise you will get the link of this PPT.
No, sir.
Right. Fine. So, and I will keep on telling you that the from which part the questions are coming, and when when I will finish this chapter, I will show you some of the type of the question, like a glimpse of the question. 4, 5, or 10 question. We'll discuss those questions, and then you will come to know what kind of the questions are being asked in the medical examination. So that's how we will do. Right? Okay. So after the class, go through with all those topics. In case if you are facing, go through with your NCRT, go through with the PPT, and if you have any doubt or any question, note it down and discuss.